So I'm Eric Admin with the Sno-King Watershed Council, and the class this evening is Bacteriological Water Monitoring. And Sno-King Watershed Council is a all-volunteer local nonprofit organization, and our mission is to help people protect local creeks and natural areas. We're also part of Global Waterwatch, which is a larger organization that operates in a number of different countries around the world. And they're the ones that provided us the techniques and the educational materials to teach you how to do these different types of water monitoring. This is an example of a bacteriological monitoring workshop that Global Waterwatch put on in Kenya. And they've got probably, they've been active in South America, they've been active in Africa. Probably one of the biggest programs right now that they have is Global Waterwatch Mexico. So they're active around the world. They're headquartered out of Auburn University in Alabama, and they're kind of a sister program with a program called Alabama Waterwatch, which is a statewide program. And their goal in that program was to put a water monitor on every stream in Alabama. And they essentially succeeded in that goal. So they have a lot of monitors, and we'll be using some of Alabama Waterwatch training materials in our class this evening as well. Okay, our agenda for today, we're going to do a super quick review of just an overview of water monitoring. Also a quick review of concepts about water, watersheds, and water pollution. And then we're going to get deeply into Global Waterwatch techniques for bacteriological water monitoring. So last night we talked a little bit about what is water monitoring, which is regular systematic assessment of conditions in the water and around the water that will affect it. People do it for different reasons. They may want to have the information for regulatory purposes, or they may want to do it for outreach and education, or they just may want to know, is the water condition getting better or worse? It's done by people ranging from government agencies to volunteers, all sorts of people, even student groups. Typically done on streams, rivers, lakes, saltwater, basically any water body that people are concerned about. And we're going to get into the how of bacteriological water monitoring this evening. So just to review, key concept is what is a watershed. And a watershed is essentially an area of land that drains to a water body. And the way that it relates to water quality is anything that happens in that watershed is going to affect the water quality in that watershed. So essentially everything runs downhill when it rains and ends up in the water. And that's why watershed is a key concept. We also talked a little bit last night about stormwater and some of the issues with stormwater. Some people are not aware that stormwater typically ends up in our streams. So when it goes down those storm drains, that's where they end up. We didn't talk so much last night about how it differs between an urban and versus a forested setting. But essentially, if you think about it, in an urban setting, there's a lot more pavement and impervious surfaces, whereas in a forested setting, it's going to have a lot more is going to be soaked up by the trees, the plants, the soil, and the landscape before it runs off. So you'll have a lot, creeks in an urban area have a lot more, they rise a lot more quickly and they drop a lot more quickly compared to ones in a rural area. There's a number of different things that can be in it, all sorts of pollutants, and the problems that those can cause are included things like pre-spawn mortality with salmon. So those are some of the issues we have with stormwater. We also talked last night about some of the things that people can do to reduce water pollution themselves. So those include things like rain gardens, restoring buffers or the areas around a stream, picking up pet waste, fixing oil leaks, avoiding using chemicals and fertilizers in their yards, using different techniques to limit the amount of runoff from their property. Okay, let's get into the details of bacteria monitoring. So the reason why we're monitoring bacteria is really we're trying to find out, is it safe for recreational contact? Is it safe for people to get into that water? And we're concerned about it because bacteria, depending on the type of bacteria, can make people sick. Bacteria are single-celled organisms. You can't see them. They're everywhere. They're in almost every environment. They're in and on human beings. They're necessary for life. There's a lot of functions that we wouldn't be able to carry out without them. And there's a lot of things that just wouldn't happen on the planet without bacteria. They're important also because they help transfer things from one state to another. So they kind of help move energy and nutrients and other things around different parts of the living world. You can group them into a family tree. For our purposes, for Global Waterwatch, we're primarily concerned with E. coli and other coliform bacteria, but there are other types of bacteria. And one of the things about this slide is it shows a color coding of the blue and the pink. And you'll see once we go through the Global Waterwatch techniques that the methods that we use, in the end, you'll end up with colonies of bacteria that are either blue, that are E. coli, or pink, which are other coliform, and we'll use that to count how many colonies we've collected. So E. coli is indicative of fecal contamination, but it doesn't necessarily mean that there is fecal contamination. This fecal contamination in water bodies is one of the major, it's a major cause of health issues, especially in developing nations where they don't have the same kind of water treatment as we do here, or safe water supplies like we do here. In those countries, you've got a lot of sewage that's discharged into waterways without treatment, and then people use those waterways for drinking water. And there's a particular strain of E. coli that's highlighted there that is the one that is the most pathogenic that we're concerned about in particular. So bacteria can come from a number of different sources, one of which is pet waste. And so that's why you may have seen some messaging, local campaigns about trying to encourage people to pick up their pet waste. There's an example of that was done in Kirkland. And actually, I think Betsy probably knows something about this project. This is where Kirkland initiated a campaign to highlight the amount of dog waste that was being left behind in their parks. And so in this campaign, they got people to go out and essentially put little flags marking all the unpicked up dog waste, and then communicate with people about the importance of picking up dog waste. And so the goal with this campaign was to get people to pick up their dog poop. Another source of bacteriological contamination in streams can be a failing side sewer. And the side sewer is something that connects a person's house to the main sewer line. And on houses that were built a long time ago, these are basically a series of concrete pipes that nest into each other. And over time, you can get tree roots growing into them. They can crack, they can break, they can start leaking, and that can make its way into the water. So that's an issue, for instance, in a watershed in Seattle, the Thornton Creek watershed. That's one of the reasons, or what they're hypothesizing, that's one of the reasons why they have so much bacteria issues in that watershed. Livestock that are not fenced out of creeks can be another source of bacteriological contamination in creeks. Another one can be wild animals. This is an example in Bothell of the UW-Bothell crow rookery. And this is a place where tens of thousands of crows roost every night. And so if you go there, it's actually really interesting to see all these crows flying around. But if you measure the bacteria levels immediately upstream of the rookery on North Creek and downstream, you'll find that the bacteria levels are uh, a lot higher downstream. Another thing that can be associated with high bacteria levels is when you have a high turbidity event. So you have a lot of rain and then a lot of stuff runs off into a creek. And this is typically happens more in an urban creek. And what we found is that when you measure the bacteria in one of these high turbidity events, that you find high bacteria associated with high turbidity. And this is an example, I think, that I showed you last night of a graph of a particular day where on that high turbidity event, the bacteria levels that we found were really high. Okay, so we've talked about some sources of bacteria. Now we're going to talk about how do you measure the bacteria. So Global Waterwatch technique uses some simple basic techniques and principles. The principle is do it the same way, do it at the same place and the same time. So you may collect at a given site monthly. There's simple, relatively low cost methods that they teach us to use. By being taught how to use these properly and using them consistently, according to the QA plan, we can generate data that's credible data. And our saying is having no information is better than having wrong information. So if you think you didn't do something properly during your testing, do the test over again. So let's talk about a concept known as colony forming units. So this is an example of a petri dish where a sample of water has been collected and then it's been placed in some media initially, which is like, and I'll show you what that is, but that's like essentially food for bacteria, and then placed into a petri dish where it's got a coating on it that will cause it to gel. Then it's incubated and you grow the colonies until they get big enough that you can see them. And when they grow and they're big enough that you can see them, then those are considered colony forming units. So those are the units that we measure. Those are the things that we count when we measure bacteria. So it's a big enough group of cells that it forms a colony and you can see it. The standard, which is used for measuring, is the number of colony forming units in a 100 milliliter sample. That's kind of the standard that's used by agencies for communicating how high the bacteria level is. Global Waterwatch teaches us a certain standard that they use for their programs internationally, and their rough guidelines are either greater than 600 is dangerous, 200 to 600 is be careful, and less than 200 they consider to be generally safe for frequent contact. In Washington State, we have stricter standards, and so they recently changed these within the last year or so. They used to be a number of different levels that they established, but essentially now for recreational use, they're saying the level is they want it to be less than 100 CFUs per 100 milliliter. A couple other concepts that are related to bacteria and bacteria levels are the concept of streams being listed as impaired. And there's some kind of technical terms that are associated with that I want to give these to you just for familiarity. It's not critical that you remember these, but under the Clean Water Act, Section 303D, a monitoring entity can send data to the EPA, or it's administered by the Department of Ecology in Washington State, saying, we think this water body is polluted and here's what the levels are of this pollutant. And so you can list a water body as polluted for any of a number of variables, including bacteria. And what happens when that listing is accepted and the water body is put on the 303D list, is the jurisdiction that is in charge of that water body comes up with a water quality improvement plan, and then they set maximum levels that they want to try to get below, and they do regular monitoring to see how their levels are. So the TMDL is the concept of a total maximum daily load, that you don't want to find the level of that pollutant above that amount. So they implement measures to try to improve water quality to try to get the levels below that. Maybe at the end, Tracy from Kirkland could comment on that, because she probably has even more information that she could share about that. Okay, let's talk about the actual steps of bacteria monitoring. So first thing is get your materials together. So these are the different materials that we're going to use for the monitoring that we do. So I mentioned there's these things called Petri dishes, and those are these little plastic round dishes that have a coating so that the sample that we pour in will gel up. Then there's what we call media or medium, and the media is essentially like a food for the bacteria. So we collect our water sample, we put it in with that media, and that's what we will then put into the Petri dish to incubate. We collect our samples with what's called a pipette, which is like a little something that you can suck up a little sample of water. And so we'll use that, a sterile pipette, so that we're not introducing any other bacteria or contaminants from anywhere else. One of the things that we measure is water temperature. So we need to bring a thermometer. And usually we do this monitoring side by side with our water chemistry monitoring. So you'll have a thermometer there with you. And one of the reasons we measure water temperature is that bacteria grows better in warmer temperatures. So usually we'll find a correlation between the lower temperatures and fewer bacteria. And then we use an incubator. And this is just the particular incubator that we've been using. It's actually an egg incubator, but what's nice about it is you can just program in a temperature. It has a thermostat, and so you can set the appropriate temperature that we want to incubate our sample at. So first thing we do before we monitor is get all our materials together. And the media we normally keep frozen, so you have to thaw out however many we want. And normally we do, for each site that we monitor, we do what we call 3 replicates. So we'll have, for each sample that we're going to gather, for each site, I should say, we're going to collect 3 samples. So we need 3 bottles of media, 3 Petri dishes, but you can use 1 pipette for all of your samples. Also going to bring our data sheet, so we can record our field observations, and then subsequently we can record our bacteria counts. And then this is just a little insulated lunchbox with an ice pack. You can use anything, but one of the things that is asked when we collect the bacteria is that we either plate it within 30 minutes of collection, or you can go up to 3 hours from collection if you carry it on ice. So normally we carry this little insulated lunchbox with an ice pack so that we can collect our samples and then be able to plate them later than 30 minutes from then, in case we're doing multiple sites, for instance. Okay, another thing that we're going to do is we're going to pre-label our materials. So we're going to put together these dishes, and I'll show you some training videos here in a second that are going to talk about how we do all these steps. But essentially, we're going to set these dishes up with labels and some tape so that we can just open them up and then tape them back closed and put them in the incubator. And we're also going to, if we're monitoring multiple sites, we want to be able to keep track of our different samples from our different sites. So we're just going to label everything appropriately. So there's an example of how you could label your different, if you had site number 1, samples A, B, and C, and site number 2, samples A, B, and C, how you could keep track of them so that you can document it properly when you're done. Okay, so let's talk about the process of collecting samples. So you're at the site. You're going to carefully unwrap a sterile pipette. You're going to use that to collect your sample, and you want to do that upstream from yourself if you're collecting in moving water, or try to avoid disturbing the water that you're collecting, so that you're not, because there can be bacteria in the substrate of a creek, or the sediment that's on the bottom, and so you may stir it up. So if you stir up turbidity, you could affect the result. So you want to collect your sample. You need to be below the surface but not touching the bottom. Then you're going to put it into the media bottle and cap it, and you can collect either a 1, a 2, a 3, up to a 5 milliliter sample. We'll talk about why you might want to do different size samples in a little bit. And then you bring it back and you plate it. So I think what we're going to do right now is we're going to watch a video from Alabama Waterwatch about collecting samples. So I'm going to stop sharing for a second here and then switch over to the video on collecting samples.
Welcome to the Alabama Waterwatch instructional video series. This video will show the proper method for collecting water samples used to determine the number of bacteria in the water body. This is the first step in a 3-step process. The second and third steps, plating the samples and determining results, are covered in their own separate videos. For each site being tested, you will need a cooler with ice, 3 media bottles at air temperature, a sterile pipette, and a thermometer. Label your media bottles by site and replicate. If you sample multiple sites, be sure to use separate data sheets or keep record of monitoring site and bottle numbers that are grouped in sets of 3 replicates. For example, if you have 2 sites, you can number the replicates at the first site as 1A, 1B, and 1C, and the replicates at the second site as 2A, 2B, and 2C. Upon arrival at the site, measure the air and water temperatures. For instruction on how to measure these temperatures, please watch the video called Water Chemistry, Air and Water Temperature. Once you are ready to sample, unwrap a sterile pipette from the bulb end and avoid touching the tip of the pipette with anything except the sample water. Fill the pipette with sample water. Be careful to not disturb the bottom of the stream. Carefully squeeze the bulb and expel excess water to the desired sample volume. 1 milliliter sample volume is recommended and is usually sufficient. Be sure to use the same volume for each of the 3 replicates and record volumes on data forms. Remove the cap from the media bottle and hold it without touching the inside. Squirt the sample water into the bottle of liquid media without placing your hand or face over the open bottle. Cap the bottle securely and place inside the cooler. Repeat these steps until all 3 media bottles are inoculated with water from the same spot. Keep in mind that media must be plated within 3 hours of collecting the sample and should remain on ice and out of direct sunlight at all times. Thanks for watching. If you have any comments or questions, give us a shout.
Okay, does anybody have any questions about that?
Eric, can you hear me?
Yep.
I was just wondering, there's space in each of these media bottles for up to 5 milliliters of water. Is that what they're saying?
Yeah, there is.
After you've thawed it?
Yes, the bottles are only about half full.
Okay. Okay. All right. Got it.
Okay. Okay, so that was collecting samples. The next stage, after you've collected your samples, you return back to wherever you're going to do your plating, which is typically your house, unless you have some kind of a lab available to you. So you're going to have an incubator ready. So you want to have your incubator pre-warmed, which in the case of the ones that we're using, you just have to plug it in. And the temperature range that we want to be in is 29 to 37 degrees. I should say Celsius, not Fahrenheit. We'd be freezing our media if we put it at 29 Fahrenheit. So that's Celsius, which is basically 37 Celsius is essentially body temperature. Then we're going to open the Petri dish just enough to pour the media in. You'll swirl it around just gently to cover the bottom. And because there's a sort of a calcium something coating on the bottom, you want to, oh, and somebody could, whoever is running the water, could you please mute? And then you'll place it in the incubator, being careful not to spill it or slosh it. And that's basically plating. So we're going to watch I'm going to stop sharing for a second here, and we'll watch a video on plating.
Welcome to the Alabama Waterwatch instructional video series. This video will show the proper method for plating water samples used to determine the number of bacterial colonies in a water body. This is the second step in a three-step process. The first and third steps, collecting the samples and determining results, are covered in their own separate videos. Prepare your Petri dishes by taping the top, the larger piece, to the bottom, the smaller piece, on one side of the dish. Keep the dish closed as much as possible to avoid contamination. Label the plate lids with labels that correspond to the inoculated media bottles in data form. Lift the top of the Petri dish just enough to pour the contents of its corresponding media bottle into the plate. Do not slosh the media onto the sides or lid of the plate. Record the time you plated your sample on your data form next to incubation start time. Be sure to shield the sample plates from direct light. Leave the plates undisturbed until the media has gelled, usually about 1 to 1.5 hours. After the media has gelled, use another piece of tape to secure the untaped side of the dish. To prevent condensation from dripping onto the plates from the lids, turn the plates over and place them in the incubator. Incubate plates between 29 and 37 degrees Celsius for 30 to 48 hours. Thanks for watching. If you have any comments or questions, give us a shout.
Okay, there's just so you know, there's been a couple of updates since that was made. And so one of the updates is that now, after we pour the sample into the Petri dish, we're placing it right side up into the incubator after we tape it, and then letting it gel there, then flipping it over. We found that we were having some delayed gelling with our samples. And the other thing they didn't point out is just the way that you tape it. It's handy that the second piece of tape you put on there, you make like a little tab where you double it over on itself, because ultimately later you may want to open it up for counting or for cleanup purposes. So that is plating. So then the next step.
Eric,
Yeah.
I had noticed on there that they stacked all three of them on top of each other. Is that the way it should be done, or should they be done singly?
It doesn't matter. Either one's fine.
Okay, thank you.
Yeah, I tend to leave them not stacked on top of each other just because every once in a while I might want to peek and see what's happening and see how the colonies are developing ahead. And if they're on top of each other, I can't see them all, but it won't affect the results at all.
Okay, thank you.
So we've done the plating. Next thing is incubating. And so they go in for 29 at a temperature range of 29 to 37 degrees Celsius, which we have pre-programmed into our incubator. And they need to stay in there for 30 to 48 hours. And at the end of that period, then you can, the colonies, that'll give the colonies enough time to grow and you can count your colonies. And this says optional, you may elect to turn them over. We've had different results, I think, depending on the size of the sample you use, or I'm not sure what other variables, but some cases, the reason why they flip them over is as it incubates, you may get condensation on the lid of the Petri dish, which can then drip back down on that gelled sample. And so it can kind of mess up the sample. So to avoid that, if you flip it upside down so that the gel part is on the top, that helps avoid that condensation issue. So now we've incubated, and the next thing we're going to do is count our colonies and interpret it. So we're going to determine results. So we're going to watch a quick video on how we do that, determining results.
Welcome to the Alabama Waterwatch instructional video series. This video will show the proper method for determining the results of incubated water samples. This is the third step in a three-step process. The first and second steps, collecting the samples and plating the samples, are covered in their own separate videos. Remove plates from the incubator, being careful not to spill any liquid. Be sure to record the incubation period, which is optimally between 30 and 34 hours. When counting the number of colonies, it is helpful to use a white sheet of paper to split the plates into quadrants. Count all blue to purple colonies on each plate as E. coli. Do not count very small or pinpoint colonies smaller than the period at the end of the sentence. Count or estimate the number of all pink to dark red colonies on each plate as other coliforms. Estimates of more than 100 colonies should be reported as abundant, and estimates of more than 200 colonies should be reported as too numerous to count. This sample should be recorded as too numerous to count. If colonies appear as a ring around the edge of the plate or as one large blob, count as one colony. Record the number of E. coli and other coliforms on the data form. The bacterial concentration in 100 milliliters will automatically be calculated in the Alabama Waterwatch database. To calculate the bacterial concentration by hand, divide the number of E. coli colonies counted by the sample volume, then multiply by 100. Kill the bacteria by pouring about a teaspoon of undiluted bleach into the dish. Let stand for about 15 minutes, then dispose of with household garbage. Thanks for watching. If you have any comments or questions, give us a shout. Welcome to the Alabama Waterwatch instruction.
Okay, does anybody have any questions on that?
We're wondering if you could put the sample in the compost instead of the garbage.
That you mean well, I think the issue is that there's potentially, you mean after you've killed it with the bleach? Is that what you're talking about?
Yes.
That's a good question. I haven't tried composting it, but I don't know. I don't know the answer to that question.
I would think that you would want to keep it out of your compost, just, because it is, I mean, it's a bacterial colony, and then you're inoculating it with bleach, which is a chemical that you wouldn't want in your compost either. So I think the best way to dispose of it is in your trash.
Okay. Thanks, Betsy. Okay, so that's determining results. So let's do an example of counting. So take a look at this Petri dish. So this is our incubated sample. We've pulled it out, and now there are a certain amount of colonies on there. So looking at the blue to dark purple colonies, again, you can use a grid, and you can even use a grid dividing it into 8 if you have a lot of colonies, and multiply if they're evenly distributed. It helps in particular with other coliform, but if your E. coli numbers are high, you can do that as well. So in this case, we have a sample volume of 1 milliliter, and we got 2. Looks like we have 7 E. coli colonies in the plate. So if we had a 1 milliliter sample, remember our standard is how many colony forming units do we have in 100 milliliters? So doing the math, that would mean in a 100 milliliter sample, we would have 700 E. coli colonies. So obviously that would not be safe for contact by Washington State or Global Waterwatch standards. And we're going to try to count the other coliform in the plate. And this isn't really something that we use this number for anything besides the fact that it just kind of shows that the test worked properly. But we do count and report the number of other coliform bacteria that we find. So in this case, there's a lot, but we're not counting the super small pinpoint ones. And this is a point of a little bit of subjectivity, but essentially we're trying to count the ones that look like they grew bigger than a tiny dot the size of a period. So in this case, we had 45 that were in 1/8. So that's our count for other coliform bacteria. So that's an example of how you would count. Then once we've counted, we're going to compare our results to the standards. And so for us, we're going to use Washington State standards, which are stricter. So we're going to compare it to, how would it compare to 100 CFU, and decide if it's safe or not. So here's a little bit different math example. So this case, we had a 5 milliliter sample. So in this case, our sample still grew 7 E. coli colonies, but the math is a little bit different. So dividing by 5, because we had a 5 milliliter sample, and then multiplying by 100, we come up with a number of 140. So not as bad as it was before. Still not what we would consider safe by Washington State standards. So we could do a number of other counting exercises, but I think you guys get the idea. So once we've counted, then what we're going to do is we're going to record our results. So on the form, there's a section where we can record our E. coli colonies for each of the reps that we did. And remember, we did 3 per site. So we'll record that. And then we will clean up and be done. We're actually looking, because there is some waste associated with this. Global Waterwatch is doing some research into some alternative ways of testing that don't involve so many materials. But currently this is the method that we're using. Now that we've done our sampling, we've counted our results, we've interpreted it, now we're going to do something with the data. So think about what it means, what you want to do with it. And you could look at several sites within a watershed. So maybe you're trying to pinpoint a source of bacteria. So you may be doing monitoring at different sites within the watershed and trying to figure out what's happening. So there's different ways that you can present and interpret the data. Again, you can use a graph, and this is an example of one of the graphs that is available within the Global Waterwatch system. So assuming that you're collecting your data and entering it into Global Waterwatch's system, you can look at any of the sites that you're monitoring and look at graphs of your bacteria numbers, and they'll be color coded. And currently they're color coded according to Global Waterwatch standards, but we're going to get them to put a line in there for Washington State standards, so you can use that as well. So this is an example of data that's on the Global Waterwatch website, and we use this in this particular site to highlight the results that we were getting with the city of Bothell. Another thing that you can do is you can put your information together in the form of a poster. Just another example of what people have done with data. So this is something that was done by the Lake Forest Park stream keepers. They've been monitoring streams in Lake Forest Park for a long time. And so they put together this poster, and it's just kind of graphic showing after the heavy rains, the high concentration of bacteria in a couple creeks. And then what happens after repeated rains, how the number of colonies drops, but there's still some bacteria present. Another thing that you can do is you can do a simultaneous data collection event where, let's say that you're trying to identify a source, you know that you have elevated levels somewhere. If you go throughout a watershed or along a creek and you have volunteers that go out all at the same time and collect a series of samples, you can compare those samples. And if you do that over time, you might identify a source. That's an example of this was done in the Thornton Creek watershed by Thornton Creek Alliance. They got volunteers, and they implemented a weekly monitoring process where they would go out and sample a variety of sites throughout the Thornton Creek watershed every Tuesday night at 6 p.m. And then over time, by collecting a series of results, they were able to identify a potential source, which in one case they identified actually a misconnected side sewer that was connected to a storm sewer connection from an apartment building. So that's another example of what you can do with this data. Here's another example showing a high number of E. coli colonies that were found at a particular place at Auburn University. And so because the levels were so high, they put up a warning sign. Here's an example from a workshop of Global Waterwatch in Kenya. And what they did is they drew a map of the watershed on the floor and had people collect their data and then put their maps down, or put their data sheets down on the floor. And then they translated that into a map and came up with a color-coded map showing the different bacteria levels at different places throughout the watershed. Another example of how bacteria monitoring data is used is something called the Swim Guide by Waterkeeper. And so this is data which in some cases is collected by volunteers and reported and then used to identify if beaches are, the level of bacteria is safe or not to go swimming at various beaches. And here's an example from Global Waterwatch Mexico, just showing an informational poster that they put together to communicate to the public different sites and what the levels of bacteria were they found at different sites. So all kinds of different things you can do with your data. And that's it for our presentation for this evening. So I want to just say thank you to all these different people and groups that helped us put this program together. And what I'm going to do is stop sharing the presentation, stop recording, and then see if anybody else has any more questions.